Well, okay, good morning, everyone, and happy Cinco de Mayo for those of you in south of the border here or in California itself, which is a big celebration of Mexican culture. And welcome to our last of the current academic year EM-X series. And of course, again, we have another exciting program. Just want to say that we will start again a new academic year, probably the first Monday of October, but we'll announce that in due course. And I'd like to thank our friend Yi Che, who has been helping run the program along with Jane Lee. But Yi has been setting up these Zooms for over 3 years now and been very loyal. His graduation is expected this summer. He'll be defending his thesis in July, and Wa and I will be on the committee, and we're hoping for good things for that. And if anyone has a postdoc in the United States, he's looking for a good position, and we can guarantee he's very good. So thank you very much, Yi. And so I'll pass it on to Wa, who will introduce our first speaker, Dr. Richard Leapman. Please, Wa.
Okay, good. Thank you, Bob. I am very happy today to have our speaker, Richard Leapman, to talk to us. I know Richard for many, many years, and he has been the scientific director at the National Institute of Biomedical Imaging and Bioengineering. He also heading the Institute's Laboratory of Cellular and Supermolecular Structure and Functions. Richard has a very unique background in our community. He was trained as a physicist in Cambridge and subsequently at Oxford and Cornell before he moved to NIH, where he initiates a number of very innovative imaging technology at NIH. So he's known for his development in the scanning transmission electron microscope and also the chemical analysis with the electron energy loss spectroscopy. And he deployed this technology in a number of biological area, including cell biology, neurobiology, cancer biology, and hematology. And today we are very happy that he can share with us his recent research on imaging 3D structure of entire eukaryotic cells using volume electron microscopy, its capability and physical limits. Richard, thank you very much for coming today.
Well, thank you very much. And thank you, Wa and Bob, for giving me this opportunity and this honor to participate in this series. Well, we began our interest in looking in 3D imaging of sort of bigger, thicker volumes of cells by developing a technique in our lab which was based on STEM tomography, and that's how we got into doing the 3D, and then from there, you know, as our collaborations increased, we went to serial block face SEM. So we'll talk about that, and then more recently we wanted to get better resolution, so we went to focused ion beam SEM. And I'll say I'm not going to. The one technique we do not use in our lab is the automated tape collecting ultramicrotome or ATOM, which was developed over the last many years. And I'll say just one or two words about that as well, just for completeness here. We'll talk about some applications to 3D imaging of entire eukaryotic cells and tissues at the nanoscale, and talk about the importance of isotropic resolution in X, Y, and Z, and these techniques can potentially examine all the organelles in a given cell and determine their connectivity and interactions. And it's difficult to do that. Obviously, the techniques of cryo-EM are complementary, but they really look at smaller volumes, typically, of cells. And then I'll spend some slides in the second part of the talk talking about some recent work comparing the serial block phase SEM with the fib SEM. And this has led us to develop a new understanding, at least it's new for us, and I don't think it was fully appreciated, a new understanding of why the fib SEM works so well. And it sort of involves, in a way, a material science explanation. So maybe I'd like, any questions on that would be appreciated. Let's see. Hopefully, I'm trying to see if I can't seem to advance the slides here, which is a bit odd. Let's see. Let me try to go to the other screen. This did not happen previously. Or maybe I've got some. Maybe now I see an arrow here. So does everybody see my screen okay now? I think I will use this.
Yeah, it's a bit small, but we can zoom in with our finger.
It's small? Okay. Let's see.
It looks well.
Yeah, it looks good.
Unfortunately,
it's good now.
When I've got the screen, I'm not able to see the. Oh, here, maybe I can do this. Okay, let me give this a try. Here we see. Let me go back if I can. Yeah. So importantly, this is the group in the last couple of years. So we have Maria Aronova, who's played a very important role, and we've got a postdoc, Zhuang, and a number of post-backs who actually spend a year or two at the NIH, and then often go to medical school or pursue a PhD. So they played a very important role in everything that goes on in the lab. Now, I should say that in 2 years ago, Nature came up with the 7 technologies to watch in 2023, and volume electron microscopy was one of them, along with single molecule protein sequencing in the James Webb Space Telescope, and a total of 7. And sort of it in this short survey of these important technologies to watch, it was mentioned. They spoke to Lucy Collinson in the Francis Crick Institute in London, and they who explains that it can take many sections of a cell, of eukaryotic cell, to look at the entire volume, and talks about the advantages of doing these imaging. And each of these techniques that we'll talk about has its advantages and also some limitations. So we will look at this here. These are just simplified schematic diagrams of the techniques that we've used. In STEM tomography, we focus an electron probe into a thick specimen and tilt it and obtain an axial bright field detector signal, which is not what you typically do in STEM, but this is what works well for looking at thicker sections. And that gives us that can give us 3D information. Then there's serial block face SEM, and this technique was developed specifically for biological specimens, really, and this is soft material, and this was done in the Max Planck Institute by Denk. Here, one uses, instead of a 300 kilovolt electron beam in the stem, down to a 1.5 kV beam. And the work is done in an SEM with a built-in microtome built into the stage. And thin sections are cut off, and you can build up, by collecting the backscattered electron signal, you can build up a volume in that way. And then, similar to this, you can instead of using a diamond knife to cut off thin layers of the sample, you can use an ion beam to mill down the sample a few nanometers at a time, and then collect the images that way. And that's the focused ion beam FIB-SEM approach, also using a 1.5 kV electron beam. And then in Harvard, this atom technique, which is sort of technically challenging to be able to cut serial sections, essentially, put them onto a tape, which goes right into a microtome, and that tape collects literally thousands and thousands of sections, sort of all lined up, and then they get deposited onto a silicon wafer. And that silicon wafer, which can have thousands of sections on it, and thousands, and maybe 100 different wafers altogether to get an entire volume, and that is imaged in an SEM with a large field of view. One thing that this requires, really, it's not in this diagram, but you really do need a multi-beam SEM in order to do this, because there are just too many voxels or pixels to collect. And so again, so let me say here that in the STEM tomography, you can get small pixel size down to, say, 3 nanometers, but you can only look at thicknesses of about 1 to 2 microns, and image fields of typically 10 microns, so small volumes. In the serial block face, you can get large volumes, but and in X and Y, you can get, say, 5 nanometer pixels. But your voxel size in the Z dimension is limited by the section thickness that you can cut off, and that is 25 nanometers is probably the best you can do, and more typically perhaps 50 nanometers. And you can look at you can collect quite a lot of data this way, and look at samples that are even up to 0.5 millimeter in size. In the focused ion beam FIB, the FIB-SEM, the voxel size is 5 nanometers, but now in X, Y, and Z, you can remove very thin layers, and but you're limited to a total volume dimensions of about 50 microns, and maybe even a little less usually. And in the case of the atom, which we're not going to be talking about here, you can get enormous volumes potentially. And in fact, it was demonstrated recently that in the last year or so that you could get up to a peta voxel size and of a 1 cubic millimeter volume of a human brain. And so these arrows just point to the sort of progression in our own lab, starting with STEM tomography, going to the serial block face, and then going to the focused ion beam. But I think you know, what the advantage of doing it that way is that we've sort of noticed some things which maybe one wouldn't notice if you just went into say one of these techniques like serial block face or focused ion beam without sort of being able to compare the techniques. And one of them is, why does the FIB-SEM even work so well for imaging stained plastic embedded biological specimens? Now, the specimen preparation is always important. We are talking here in these machines about sort of fixed and stained preparations. So we don't quite have the native structure, but we do have the advantage of the whole cell, and that is quite important as well. And so STEM tomography, you can use a somewhat light amount of stain, but special techniques were developed for the serial block face and the FIB-SEM because the signal really does depend on how many of those stain atoms you have. And so some variations were developed by Mark Ellisman's lab at UCSD, and that included the addition of some reagents here and putting additional amounts of urinal and lead into the specimen. Samples can also be prepared by rapid freezing and freeze substitution. So with that we've made use of for some of our work because when we were concerned about the possible artifacts due to chemical fixation. Now, just very quickly, the basis for STEM tomography. Why do STEM? Well, in the TEM, when you're looking at a thin specimen, most of the electrons are scattered, go right through the sample, and there's very little inelastic scattering. That's shown here in the energy loss spectrum. There's some amount of inelastic scattering, but not an awful lot. So everything is focused for a thin specimen. The image is focused in the TEM. But when you make a thick specimen, so if you make the specimen 1 micron thick, there's a huge amount of inelastic scattering shown in the energy loss spectrum, and just a tiny little blip of the elastic peak is a nonlinear effect, obviously. And that big spread of energy loss means that the image is blurred, and that's what you see when you look in the electron microscope at a thick 1 micron thick biological specimen. Everything is incredibly blurred. But in the STEM, there are no imaging lenses below the specimen, so you form a very narrow, a very focused probe on the specimen, and then instead of using a dark field detector, you look at the sort of the absorption of the beam, and you have a small axial detector. That of course is diffraction limited, but when you're only after a nanometer or a few nanometer resolution, which we are in these cases, it doesn't matter if you've got a small angle in the scattering. So that is how that works. And then this is an example of something that we did a number of years ago, but we have now taken this up again, and we're getting new data from the same system to ask some different questions. Here we see a 1.2 micron thick section of rat retina showing a structure known as the ribbon synapse in rod bipolar cells. And the important thing is that this will become evident in the center of the image here. And this can be segmented here. But the important thing is these are very sparsely distributed synapses. They're not everywhere. You have to sort of search around it, but they're very, very important structures. And in order to see how these things what they look like, you really need to have a 3D picture, and you need to be able to find them as well. So the analysis was done. This is a sort of schematic of the laminar organization of the retina, and it's about 200 microns thick in the rat. And the features that we're seeing in these tomograms are just in this very, very thin outer plexiform layer of the retina. Where synapses are formed between the bipolar cells and the rod cells and other cells that are coming together in this region, the horizontal cells there. And so from these images, one can notice that you see little tethers for the vesicles that are anchored to the plasma membrane of the bipolar cell, and you can measure. These are the docked or tethered vesicles, and you can measure the length and height and width of this ribbon. And this ribbon, our collaborators in the Jeff Diamond lab at NIH, they believe that this model of the docked vesicles is a way that the cell has found to be able to concentrate these vesicles so that they're used when they're needed in these and can release the vesicles. And there's always a releasable pool anchored to the plasma membrane, with others that are on their way down to take the place of the ones that have been released. So that was sort of an interesting project where I think it demonstrates that you need. I mean, this was not done with this was done with the STEM tomography, but it nevertheless was a thick volume compared with what one would normally do in tomography, electron tomography, for example, where one would only be looking at a sample that maybe 200 or 300 nanometers thick, and where you would miss certain features of this structure. Now, another application that we worked on, have been working on for a long time, was sort of a system that is both important, but also has the advantage of being somewhat simple in terms of the structures. And these are the blood platelets. We've looked at both human blood platelets and also animal mouse platelets. And in this case, again with STEM tomography, you're limited. You don't see the whole platelet, because these are a few microns in size. But nevertheless, you can get a good representative slice of the plate like structure, and you can see the organelles. There is the blue organelles here are the alpha granules, which contain a lot of compounds that are released by these cells and participate in the blood clotting cascade. And then there are mitochondria in purple here. There is a few dense core granules in red, and large amount of membrane stored in the so-called canalicular system. And when we looked, and you can see this a little bit clearly here, when you activate these blood platelets, and that activation could be as simple as just drawing the blood. So you have to be really quite careful about making the samples. But when they are resting, you see smooth membranes, and when they're activated, you see these holes appear in the membrane. And those holes, if you look at the inside, they are connected to these alpha granules, which are forming necks to the plasma membrane, and they release their contents through those holes. So that was an interesting observation. Now, the scale of biological structures, you know, what we really would like to look at is whole eukaryotic cells. And blood platelets are eukaryotic cells. They don't have a nucleus, and because they've been they're sort of being produced constantly. They don't need a nucleus, and also they're very small. The typical eukaryotic cell is 10 or even can be bigger microns in size, and that cannot really be done by STEM tomography. And so this led us to and the interest in the types of collaborations we're working on. It led us to try to use other volume electron microscopy techniques. And we started with the with a Sigma Zeiss Sigma instrument, which had a Gatan 3-view system, which was the microtome that was in the specimen stage of the microscope. And that's the picture of the instrument there. And you can see the microtome inside the column, and a section is cut off the block, which is situated as shown, and the beam is scanned, subsequently scanned with the backscatter detector collects a signal from the specimen, and then that's repeated until you get a whole volume. So now you can see in these activated platelets, you can see the whole volume. So it doesn't quite have the resolution of the STEM tomography, but you can see nevertheless the structures, and that's shown here. Now, one thing I should point out is a key thing. We've done a little bit of work in this area, but it's essential. There's so much data that one's collecting that to make any sense of it, you really have to do some segmentation. And the segmentation, fortunately, has been progressing, you know, with the deep learning techniques that can be used. And a few years ago, we had a postdoc in our lab and a graduate student, Matt Gay and Ziad Imam, and they developed a technique based on the sort of standard UNET, but had some other ideas about how to make this work. And they succeeded in being able to segment these platelets. It's sort of a model system in our hands. There's a limited number of organelles, and they were able to segment all the key organelles in the cell. And then we sort of looked at the quantitative analysis of the results that came out compared with what an expert annotator would obtain, and we got quite good agreement using the automated segmentation. And then more recently, just with our current postbacks, everybody now, a lot of our postbacks are very interested in the machine learning and AI, and it's amazing how adept they are at doing this just after an undergraduate degree. And so they launched into this and developed a slightly different approach still based on UNETs, but nevertheless, you know, with their own ideas about how to segment another aspect of these blood platelets, and these are blood clots. And so that's our collaborator, Brian Story, in the University of Arkansas. He is interested in understanding the structure of the blood clot, and that's so we've looked on the left here, the top left, you see a cross section of I think this is a jugular vein, but I'm not absolutely sure what the blood vessel is. And it's occluded by a blood clot. And what is interesting is that when you look at the segmentation, there's thousands and thousands of cells here, and each one has to be segmented. And so you have to come across some sort of automation. But what's interesting is you see different regions in the blood clot. It's not just it's not just homogeneous throughout. It's a heterogeneous structure, and you can even see in the way they've segmented it. You can see the mitochondria and alpha granules within these individual cells, and the cells are all crammed together. So that's one thing that we are working on, still working on, and but it shows how you need to look at a volume. Now coming back to another problem here, which we looked at, we published this a couple of years ago, 4 years ago. The identification of immature secretory granules in mouse pancreatic islet. Now, this is a seroblastase SEM image going through an individual part of an individual beta cell. And what we'll see in this movie here is that in this region in the center of the image right now, close to the nucleus, is some Golgi membranes. And in those Golgi membranes, you see a bunch of these immature secretory granules, which look quite different from the mature granules. The mature granules have these halos around them, and this we'll see a little bit more. Let's see. Yeah, so this is a bit clearer here in the sort of a single slice of this. And if you look in the little insets on the left of the image, you'll see these immature granules, which do not have halos. They look quite smooth, but they're well defined, and they have membranes that are about the same size as the mature granules with the halos. And that's also shown on the image on the right, which shows the region within in the Golgi area, where you can see a whole group of immature granules there, and that from that we were able to analyze the total volume of the contents of these granules. They're jam-packed with proinsulin, the immature granules, and the mature granules have just insulin. And so you can measure the ratio of immature insulin to proinsulin to insulin in the cell by adding up all the volumes of all of these granules, and that enables, and given one or two sort of other points that we know from the biochemistry, like for example, you know how much insulin is released from the cell per unit time. We can, from those, using a radioactive label, so you can calculate what the maturation time of the immature granule is, and that's shown here. It turns out to be about 135 plus or minus 14 minutes, which sort of is an interesting idea that one can get some dynamic information from a volume EM if you can identify the particular structures of interest. And so we did that. Now we moved to the Fib SEM, and here you can see this is the cross section of the Zeiss 550 that we have, and with the focused ion beam coming in at an angle, you tilt the sample to that angle so that it's perpendicular to the fib beam, and then you collect the data. And and what you see in the X and Y and in the XZ, for example, the images look practically the same. That is not the case for the serial block face imaging. There's a much worse resolution in the Z direction. And but one thing that we did notice we'd done a study a number of years earlier where we did some Monte Carlo simulations of the scattering, and what we found was that at 1.5 kV, because we were interested in seeing how far down does the information come from how far down in the block does the information come from when you're looking at 1.5 kV structures within the block. And it turns out that if you look at this 1.5 kV sort of histogram of simulated stain at depths going down from down 0 to 12.5, 12.5 to 25 nanometers, all the way down to 50 nanometers. The first two blocks there, that is the blue and the red, they're almost the same height. So, you shouldn't be able to get better depth resolution than 25 nanometers at the best in the FIB-SEM. But that is not what we see. We see, down to sub 10 nanometer resolution. Why is that? And that's one of the mysteries which nobody was able to answer. I asked 100 people who were supposed to be experts, and nobody could figure that one out. So again, you see this better resolution than you should expect in the Z direction. And so the geometry of the specimen block this just shows the geometry here. So what you do for the biological specimen is you've got an epoxy block. You have to cut a trench in front of the block face. The block face is shown. It's along the direction of the gallium beam, and you have to dig this trench because you have to get the electrons out. If you don't have the trench, you can't get the electrons out when you try to image it with the electron beam. So typical operating conditions for the FIB-SEM: 30 kV gallium ions with about 700 picoamps for imaging, 1.5 kV electrons with a current of 2 nanoamps for imaging, pixel dwell time of about 8 microseconds, pixel size down to 5 nanometers, signals collected with an in-lens energy-selected backscatter detector. Turns out that the energy selection in our hands, it doesn't make any difference, basically. Even though people said it was important to do the energy selection, it doesn't really help. And then the gallium ion and electron beams operate simultaneously. So the electron fluence is 4,000 electrons per square nanometer, and yet the sample does not collapse. Compare that with exactly the same specimen, exactly the same preparation, in the scanning, in the serial block face SEM. And there you've got 1.5 kV electrons at a current of about 40 picoamps, pixel dwell time of 2 microseconds, pixel size of 5 nanometers again. That gives you an electron fluence of 20 electrons per square nanometer. And if you go higher than 20, suddenly if you double it to 40, you get specimen collapse. So for some reason, no one had questioned this result, and the discrepancy was ignored. Why is that the case? So this is now some recent work on, let's see. So this shows a 3D data set from cultured rat hippocampal neurons acquired in the crossbeam at a beam energy of 1.5 kV. And there you can see the individual secretory vesicles with their which are 40 nanometers in diameter. And then, so this is just a cross section there. You can see a nice lumen inside each vesicle, obviously so other structures in this. This is a single slice here just to show this, but then the rendering shown there. So we were puzzled, and so what we did is why is there this discrepancy? So we took our block out of our fib block, where you can see the trench here, or trenches, because we imaged a few different places here. And here's the block face, the sort of the edge of the block face. And so we imaged this. We cut off the protective platinum pad that's laid down on top of the block, and we imaged the block face in the fib in the serial block face SEM, and you can see here there's a bright line right at the which is maybe not that surprising. I mean, there's some gallium that's being deposited. What's interesting is the gallium, when we cut it, it's a little bit fragmented. You can see signs of fracture right at that edge, and it's about 20 nanometers in thickness. Now, when we then took the.
Hi, Richard. Can you wrap that up in a couple of minutes? Yeah. 1 minute.
1 minute. So
okay,
we took the edge of we put it into a microtome, and we cut sections, and that's shown here. Now there's a really bright edge, and then when you look at the elastic scattering, that's an elastic scattering high angle dark field. We put this in the STEM to analyze it, and from quantifying, knowing the cross sections, scattering cross sections for the gallium and the carbon, we can predict that the layer contains between 25 and 50% atomic percent gallium, and that's also shown when we fib mill an EPON section. Again, you see that layer. And you can actually detect the gallium there. This is the gallium. This is the EEL spectrum from that edge, and you can see the gallium signal and the carbon. And then we looked at this. We found this paper which had been written in 2021, which used gallium fib milling of a gallium fib milling of EPON to build microstructures, cantilevers for atomic force microscopy. And this paper has only been referenced by the polymer scientists, and nobody in the field of fib SEM in biological specimens, volume EM, has noticed this paper. And they show that the Young's modulus of this thin, stiffened layer was calculated to be 30 to 100 gigapascals. And then we were able to do simulations here. So we have this gallium. We could do the simulations with no gallium, with 25% and with 40% gallium, and we find that the information, the gallium prevents it from for the electron beam from going far into the block, and so that's why we get the better resolution. And we were able to do Monte Carlo simulations as well. I think this is the last slide, showing that without if you do a simulation without gallium and you get a blurred out secretary, this is a secretary vesicle, you get a blurred out structure. When if you do it with the gallium, you see what we see in our images. You see a sort of 40 nanometer sphere there. So in summary, a variety of volume EM techniques are available for application to biological systems. The ones I've talked about, each has some unique advantages and limitations. The mechanism by which fib SEM provides 3D data with high depth resolution and high signal to noise is just becoming known. The gallium creates a conducting material, I think a glassy phase that covers the block and prevents charge buildup. Because it's metal. The surface gallium-rich layer created in the gallium fib helps protect the EPON from damage because the incident electrons do not penetrate deeply into the block. It also improves the Z resolution of the fib SEM relative to other volume techniques. So with that, I thank you very much, and if there's time, I'll try to answer a question.
Okay, Richard, thank you very much. This is fascinating talk. And I think there's a lot to be learned yet in this bottom EM in biological samples in understand the physics behind it. I think your lecture definitely illustrates the importance of the collaboration between the physicists and the biologists. In the interest of time, I would suggest that we open your lecture to questions after the next speaker. So if you can stay around for the
Sure,
next
absolutely.
lectures. Okay, and then
I'll stop sharing here.
Yeah, right. Good, thank you. And I would turn the floor back to Bob.
Good. Thank you very much, Wa. Thank you, Richard. That was fascinating stuff. And yes, I agree with what Wa said about how this combination between biological and physical sciences is really another avenue for exploring the applications of electron microscopy in both our fields. We've learned a lot for our specimen preparation from you, as well as many of the biological applications of physical science electron microscopy as well. I'd like now to introduce Kate, who has agreed kindly to host Vesna Srot from Germany, the great Stuttgart lab. And so, Kate, I'll hand it over to you to introduce Vesna, please.
Yeah, thank you so much, Bob. And it's my pleasure today to introduce Dr. Vesna Srot, who is currently a research scientist at the Max Planck Institute for Solid State Research in Stuttgart, Germany. Before that, she obtained her PhD in geology and mineralogy from the University of Ljubljana, Slovenia. And she has a very broad range of research interests, including sample preparation and stem characterization of hybrid organic, inorganic, biological, and bio-inspired materials, as well as method development for MEMS-based in situ stem investigation of functional materials by electrical biasing and electrothermal experiments. So as someone who regularly does in situ sample prep, I've been closely following her work, and I'm very excited to learn more from her today. So thank you very much for speaking with us, Dr. Srot.
Thank you very much. Good morning to everybody. Thank you for this kind introduction, and thank you also to organizers to giving me this great opportunity to show some of my work today. So as you can see from my title, I will talk about in situ sample preparation and about the characterization of these samples then in TEM. So we know that TEM offers high spatial, high temporal, and high energy resolution that provides unique information then for understanding properties and functions of materials. With TEM, we can characterize morphology, chemistry, crystal structure, electronic structure, and these can all be used for many different applications. Here are just some of them, so such as catalysis, electronic devices, energy materials, in material science, for nanomaterials, and in life science. Under controlled environmental conditions and with the ability that we can dynamically influence our samples by external stimuli, we can now turn our microscopes into a powerful laboratories. And due to this recent, very quick development of specimen holders and introduction of integration of MEMS devices, we can now apply also in TEM external stimuli such as biasing, heating, cooling, liquid, gas, light. However, the validity of our data is very strongly dependent on the quality of our samples, because always when we prepare the samples, even when we thin them down to electron transparency, they still must have these pristine properties. Otherwise, we failed our job. There are different sample preparation approaches for in situ electrical and electrothermal experiments. However, most of them, they include this intermediate fixation to another FIB grid or to something else, and only then they detach it and again attach it to the chip. Just recently, a Protochips company developed so-called FIB optimized chip, and they use it in combination with this pre-tilted holder. Just to show you what is that we will all be on the same page, what is the difference between this newly developed FIB-optimized E-chip and other electrical electrothermal chips. So previously electrothermal chips, they have all the membrane, and then we have these electrodes that are printed directly on the membrane. And when we land, when we deposit our lamella, then when it's deposited, we have access only from the top side, because from the bottom side now we have a membrane. We cannot do anything from the bottom side. However, in the case of FIB optimized chip, we don't have any membrane, so here is a hole. The electrodes are printed directly on this silicon wafer, which means that we have now easier access to the lamella front and back side, and here I should write also from the top side. What was the motivation for our work, and what is still motivation for my continuous work? So that we prepare clean and artifact-free samples. This is also for normal preparation, not just for in situ. However, when we are doing electrical measurements, that samples are clean is even more important. I also wanted to achieve reproducible results, so it doesn't help me if I can prepare only one lamella and this works, and then the second one will not work anymore. My great wish was also to reduce, or at least to minimize, these attachment-detachment steps, because if I manage to do this, I will minimize use of platinum. And if I can minimize use of platinum, I will also be able to reduce contamination of platinum and gallium. So today I will show you detailed step-by-step preparation of in situ samples using new optimized methodology and also characterization of these samples. And if you want to follow, or if you want to prepare it by yourself, you can find detailed steps here in this manuscript. What are the requirements? So since it's FIB-based, we need FIB, and this FIB needs to be equipped with micromanipulator needle. When we do FIB sample preparation, a lot of people, they like to work with some fat, ugly shaped needles. In this case, now we have to really shape the needle that it looks like a pencil. That's very important. And when it comes to holders, it's very simple. So if you will prepare chip, if we will prepare lamella on the chip with a membrane, you just need two classical, these flat holders. In the case that you will work with FIB-optimized chips, you need one holder where you will place your sample. And this is the holder with manual tilt possibility where you will put your chip. So what is the main difference when we of this new method compared to previous methods? So for classical FIB, we do also pre-cutting and we prepare U-cut. In the case of this new method, we must leave extra space on one side of the platinum layer, of the platinum strap, because you will see later why we need this. And when we prepare normal samples, we will retract, we will take lamella out in this orientation. In my case, I use alternative orientation during the lift-out procedure, so I rotate the stage for 90 degrees, because in such case, I can now deposit lamella directly to the chip. I do not need intermediate step to deposit it on some other grid. So I used FEI SIOS system, And intentionally, I decided for the most boring sample that you can use, that's silicon, because I started with some complicated samples and I didn't understand anything because there was contamination and I didn't know whether it's contamination or it comes from the sample. So that's why I decided to work with silicon, because then there will be also, when I apply current, there will be not much changes. And also I will immediately see if there is contamination. So procedure is the following. First, we deposit platinum protective layer on the area of interest. And as I said, it's important that we leave on one side a little bit more space. So in general, I do a little bit bigger pre-cuts because then it's much easier to remove the lamella. It will take a couple of minutes more. However, you will be able to take lamella out. Otherwise, you might lose your lamella. Then we do the U-cutting. Here it's a little bit different. So with U-cut, I go a little bit higher. However, we should not go too high so that the lamella still stays stable. Then this is the important step. We rotate our stage together with the sample for 90 degrees in clockwise direction. And now we insert our nicely shaped lift-out needle and we fix it here at the top side. And in the next step, we cut our lamella free from the bulk. This step is especially at the beginning a little bit critical because it's hard to see when lamella is really cut. That's why I recommend you, when you do this step, that you try to retract lamella very slowly, because if it's not completely cut, you still have a possibility to rescue it. Maybe you can continue with cutting. Then we retract, we take lamella out, we retract the needle. And at this stage, I would highly recommend to lower the stage, because in the next steps, we will do a little bit rotation, a little bit gymnastics with the needle, and it's always better to have a little bit extra space. So our stage is now lowered, and now we again insert our needle together with the lamella, and we rotate it for 90 degrees in anti-clockwise direction. In the case that you will use chip with a membrane, now it's important to clean. This will be your backside. So this will be the side that will sit on the membrane. It's important to clean now the backside because later on you will not have any more possibility to do this. If you will use FIP optimized chip, you don't need to do here anything, because also later on you will have access to the backside of the lamella. And then we rotate for additional 90 degrees in anti-clockwise direction, and now we are ready to land. Now we have to do one more step. We again retract the lamella, because only at this point I insert the chip in the chamber, because I do not want to have the chip all the time in the chamber, because we would just unnecessarily contaminate it. And then, so this, what you can see here, are 4 electrodes on FIP optimized chip, and then we slowly approach with the lamella to the chip. And here it's a little bit you have to get used to. I always swap between SEM and FIP mode because I observe the shadows so that I don't crash with the lamella into the chip, so that we gently land the lamella. As you can see here with this method, we can really nicely land it on full contact. Then once when it's landed, I usually first deposit one contact here just diagonally from the needle. And then we cut away the needle and we fix also other 4 contacts. In the next step, we have to cut the edges. The top edge we must cut, because otherwise this platinum layer, it would be conductive between these 2 contacts. The bottom side you don't have to cut, but I would strongly recommend you to cut it. I will show you later on why I recommend that. Then, so we performed this landing when chip was in horizontal orientation. Now, since we have this holder with pre-tilt possibility, we don't need now to remove chip and fix it somewhere else, but we just simply remove the holder together with the chip from the chamber, and we manually tilt it. So in this case, I prefer to do vertical preparation. So I tilt it in vertical preparation, and I continue then with the preparation like we would do the normal sample preparation in vertical orientation. Why vertical preparation? If you have pre-tilted, you will be able to see only the front side of the lamella. The back side you can thin it and do whatever you want, but you will not see it. In the case of vertical preparation, if we just rotate the sample for 180 degrees, we are able to see, so this is back side, a lamella from the back side and lamella from the front side. And now back to the shaping of the lift-out needles. So if you are using these potato-shaped needles, you will not be really able to nicely fix the lamella to the needle. That's why I would recommend that you shape the needle so that you leave thickness that it's approximately of the same thickness as the thickness of the lamella. And if it's very thick, you see, you also block your field of view. And now I can show you why I cut also the bottom side, so the bottom edges, because if lamella is massively contaminated, like here, we will see this. But sometimes we have only slight contamination, as you will see here. However, I measured several times that also very minor contamination can change electrical properties. And that's why especially the bottom side, it's not always easy to clean. That's why please do the cut. I always do double cut, which is not necessary. Single cut would be enough, but just better safe than sorry, I do double cut. And now to the holders. So this is a double tilt protochips fusion holder. I just put here in the holder, so this is how it looks like when normal chip with the membrane is inside, and this is how it looks like when we have FIP optimized chip. So you see it's approximately half of the size, like you would cut this chip in half. And here, so this is the tip of this holder, and you can see we put chip here and this dot here. So our lamella, here we see the electrodes are running here towards this hole, and here is our lamella placed. So the setup at TM, in all the samples that I prepared and also intermediate steps, I checked in TM after each step. When I did some thinning, cutting, whatever, I always checked in TM what is happening. Samples were investigated using 2G ARM microscope. This one is image corrected. This one is probe corrected. Samples were investigated using 200 kilovolts in TEM and STEM mode. All samples were characterized also with EDX, and the ones that were thin enough also with EELs. And I used also, because in our case, we have single and double tilt electrical biasing holder. So this, what you see here, it looks a little bit ugly, but I noticed that it really helps when we fix cables with tape. So doesn't look nice, but it's efficient. All samples were measured by using 4-point measurement technique. I applied many different currents, going from very low currents, but also to going to very high currents, because I wanted to see how stable my contacts are. I wanted to see at which point I will break the sample. All measurements were done in 3-step waveform mode, so I applied current for 600 seconds. I was holding the current for 200 seconds and then reducing the current for 600 seconds. I also used alternative timing, so from very short times, like 1 minute per step, but to a very long timing, so 1 hour per step. And I did not, so for this case of silicon, I did not notice major differences in this case. Then I was wondering, I wanted to know what happens if we change size, if we have like small contacts or big contacts, if this will make any difference. So first, I deposited contacts for this lamella only, so from the side. The contact was touching lamella only from the side. I performed current voltage characteristics, and then I put the same sample back to FIP, and then I extended the contacts so that they were extending over the surface of the sample. And we see here that there is no major difference in the resistance. However, what we notice that there is superior stability and reproducibility if platinum contacts are extending over the lamella surface, especially when we use higher currents. It is known from the literature that when we deposit platinum contacts at different voltages, this might change the composition of the contact. So there will be difference in the ratio between platinum, gallium, and carbon. That's why I decided I wanted to see what will happen if I deposit contacts at 8 keV and at 30 keV. So this upper side here, contacts were deposited at 8 keV. Here, bottom images at 30 keV. What we can see immediately is that these contacts deposited at 30 keV are sharper, and that contamination around the lamella is much smaller when we have contacts deposited at 30 keV. At 8 keV, we create much bigger halo around the sample. Then we also at each step also I measured current voltage characteristics first when beam in the microscope was off, then with beam on, because also when we put beam on, we might change the characteristics of the contacts. What we notice at the end, so here current voltage characteristics show that sample where we use deposition conditions at 8 keV is slightly more conductive. And we interpreted this is most likely due to this massive contamination around the sample. Then, when I started with in situ, I asked several people, how thick should my lamella be when I prepare it? So how thick should I cut it? And nobody was able to give me any answer. They just said, should not be too thin, because then when you land it, probably it will break, so it's mechanically unstable, but also not too thick, because then it will be hard to fix it. So what I did, I prepared 3 lamellas using the same conditions. Only difference here is the thickness. So one lamella was 1 micrometer thick, then 1.5, and 2 micrometer thick lamella. Then I performed current voltage characteristics for all 3, and we can see that while applying the same current of 100 microamps, this results for the thickest lamella, it results in the lowest voltage. And we can see also that for the thickest lamella, there is almost no variations in the resistance. This is most likely due to the biggest volume and the biggest area cross-section. Then we have studied current voltage characteristics of the lamella that was designed in a specific way. So first, we thinned uniformly the middle part of this lamella, as you can see here. And then we have started to form a notch from the bottom side of the lamella in a stepwise manner. So first we left here 8 micrometers of material left, then 4, then 3 and 2, and at the end also sample was additionally thinned. After each cutting or cleaning step, I took the sample to TM and performed the full characterization, so current voltage characteristics, also EDX, and when it was possible, also EELS. We can see that the resistance changes only for the structures where we have the most narrow width here of the sample. Now, to better understand our data and what we did here, we considered our structure to be resistors in series. So it is known that the resistance is directly proportional to length and resistivity and inversely proportional to the area cross-section. Length and width we determined from SEM images, and the thickness from low-loss EELS, so that I could also determine the area cross-section. The resistivity was determined from the experimental data that we obtained. And what I did then, we could calculate the total resistance, and this total resistance is now here plotted versus so here is the ratio between the area cross-section of this small part here that was cut and of this area cross-section of this part that was not cut. And we see similarly also for this calculated data, very similar than for our experimental data, that we see high increase in resistance only when we have the most narrow width here of the sample. Now, to show you how I check the quality of this sample. So here you can see HADF stem images acquired at lower magnifications to show you that we can obtain really huge, really large areas that are super clean. Here you can see high resolution bright field and HADF stem images. So this is really only pure FIP prepared samples, no additional cleaning in some other machines. Also, EDX shows that we have really only silicon present, so super clean samples, and also from eels, we can see that this is typical spectrum for pure silicon. So we have very clean and artifact-free surface for most of the samples. That's why each sample I always characterize also with analytical method so that I can judge the quality, and if necessary, I also improve the quality. This, what I showed you up to now, was on FIP optimized C chips, but the same quality of the sample I can obtain also when a sample is deposited on the electrical chip with a membrane. These images are a little bit worse than the ones on the previous slide, but this is also due to, because these images were acquired in image-corrected microscope, the other one in probe-corrected. I thin lamella here directly on the chip. As I said, that this method is not applicable only for silicon, but we can prepare any sample that will survive FIP preparation. So if you can do fibbing, you can use this method. Here is an example of indium selenide. So indium selenide is very sensitive for the preparation. However, this is the sample that was prepared on the chip. You see also here is super clean sample. However, life is not always as easy as one would think, and we always like to show only beautiful results, but I made a collection also of failures, because when we do in situ, this happens very often. So most of the time, we fail. The days when everything runs perfectly are not that common, but that's why we are even more happy when this happens. So just here to show you, so in the evening, I deposited lamella on the chip. And in the morning when I came, lamella was gone. I still don't know where the lamella, I know it's somewhere in the chamber, but I don't know why it's in the chamber. Then the second example, so also in the evening, I left it like that. It was fixed, it was stable, I tilted, everything was nice. In the morning, there was a bunch of garbage on that lamella, and it was so extremely contaminated that I even was not able to clean it. It was really total disaster. Then this is a case where we are still not sure whether sometimes it happens that stage jumps or the needle jumps. We still don't know what happens, but this lamella was already perfectly landed on all 4 electrodes. And then all of a sudden the needle or stage jumped, and you see now the needle is bent, and under such orientation, I could not land anymore. So I just cut away here the needle and start new. Then in this case, so this is a chip with a membrane. I deposited on one side half of the lamella, and on the other side, I was trying to deposit a needle, and I was a little bit too brave, and I punched the membrane. And of course, my needle flew away. In this case, that was quite interesting because lamella was already fixed on the electrodes, and then first it flipped a little bit. I tried to push it a little bit down, but then it completely flipped on the membrane. And once when this happens, you cannot do anything anymore. It's there forever. Then, so problems are not only in FIP, but also in TM we ran into troubles. So this was half of the lamella, and there was one specially designed needle. Already when I was preparing this, so it took me 5 days to prepare this sample. Already in FIP, I saw teeny tiny crack, but I said, it's okay, I can do this. As soon as I applied a little bit current, this just exploded. I just got a big crack here, and of course these 2 needle and the sample were not anymore in the contact, so experiment finished. In this case, it was just because of my stupidity, because this was perfect lamella. And everybody knows that when we do electrical biasing, we have to ground the sample. And I forgot to do that. So this was the 1 second experiment. I just started the current, it just exploded. I went home. It was end of the day. So to conclude, I showed you new FIB-based methodology that we can use for preparing TM lamella on MEMS-based chips. Here I wrote that it's only for in situ electrical and electrothermal, but you can use it for any. You can deposit lamella on any flat surface. It can be used also for liquid and gas cell preparation. It's suitable for any material that will survive regular FIB preparation methods. Requirements are quite minimal, so of course we need FIB, but regarding the holders, these are just very simple holders that every lab usually has it. Here I use alternative orientation for lift-out procedure, and because of this, I can deposit lamella directly on the chip. I don't need any intermediate steps. I showed you that there is minimal impact of the beam energy during platinum deposition and minimal impact of the size of platinum contacts. And I showed you that there is systematic influence of the lamella thickness on current voltage characteristics, and that also if we cut slits into the lamella, we might influence electrical characteristics. And at the end, I also showed you that we can prepare really clean samples, artifact-free, and we can have really big areas that are thin and clean. At the end, I would also like to thank co-authors on my work, Professor Dr. Van Aken, Dr. Rainer Straubinger. We did this work when he was still at Protochips, and Miss Felicitas Predel. And also I would like to thank Dr. John Damiano from Protochips, because we had several very fruitful discussions. And at the end, I would like to thank you for your kind attention.
Thank you so much, Vesna. That was incredible and really amazing how clean you can get the samples, especially for that indium selenide and the 4-point probe measurements. It was very impressive. I particularly like the Murphy's Law FIB and TM editions. I've been there a few times myself.
Yeah,
so happy to open the floor to a few questions, or do we want to do, I think we'll open the floor to a few questions now, and then we'll transition over to our other speaker for questions.
That's good, yeah. Let's ask questions for Vesna.
Yeah.
Thanks, Vesna, for the very exciting and very detailed talk. So I have maybe a naive question. So is it possible, like, when you deposit, like if you consider depositing like platinum or some other metals to connect like all these 4 electrodes, how will that change the thermal property of the specimen?
I mean, it's like this. First, I don't have any other possibility, so I can deposit platinum and carbon. However, I think for this case, because these chips, the electrodes are platinum, I think it's better that also contacts are platinum. But probably it would change, but I don't have any experience with this, but I think why platinum is also good, because I see sometimes when I land, I think it's also quite elastic. It's quite forgivable. Also, if you push a little bit, lamella will bend a little bit, but it will not immediately crash away. So that's why I like this platinum. But probably if you use some other metal, this would change the properties. Yes.
Thank you. Yeah.
I had kind of a follow-up question actually on the contacts. For some of these like indium selenide or these 2-dimensional or low contact materials, do you ever see a large contribution from contact resistance when you're fibbing that platinum onto the existing contact, or does that affect your measurements at all?
No, we are at the moment we are working exactly on this material, and I'm preparing samples in different orientations, different thicknesses. So I'm not quite sure. I think that bigger influence has if if sample is not completely thin, because I notice on the sides, I thin mainly the middle part, and we still have some leftovers, because this is also something what I discussed with some other people that are working. They say that there is still in the thicker part, maybe some leftover of gallium, that we cannot clean it. And this will influence the measurements.
I see. Okay, thanks.
So this is still something that I'm exactly working at the moment. I'm also trying to clean on the sides as much as possible. Not everything can be cleaned. I try to do my best, but I do see differences now when I'm also cleaning really between these contacts, these areas that usually nobody is cleaning. But I started now to clean also these parts.
Okay, fantastic.
Yeah,
I was going to ask a question about the, would there be any advantage in using like a plasma type of source rather than, some gas source rather than gallium, or is that not possible?
Yes, I mean, in my case, it's not possible because I have only possibility of gallium FIB, but I read in the literature so that with plasma FIB, that they can achieve better quality, so less contamination. Yes.
I think we found that the delineation is not as sharp. So with the platinum, also with the gallium, you get much flatter and sides, which you can define much more sharply than with the plasma. But maybe that's just a development problem. I don't know.
I mean, I read the papers, and of course, because they use plasma FIB, they will say that plasma FIB is the best. So I don't know. I cannot judge because I didn't use it. I can only talk about what I use it, and I know that I'm still far from. I can optimize much more, but I'm doing now step by step because it takes me. I always thought I'm a little bit faster, but with all the pre-cutting, because we have to cut a little bit bigger area, and these chips, these FIB optimized chips, the distance between the electrodes is 25 micrometers, 20 to 25 micrometers. This means that lamella has to be huge. And so I was looking at all the pre-cutting and fixing and thinning. It's around 3 days if I work around 12 hours per day. So to produce one such chip, that's why I am testing, but it takes a bit longer because first I need to make lamella and then I'm testing. But I have at the moment really multi-production. So also when I'm not downstairs, FIB is just cutting for me. I'm just doing pre-cuts in advance so that I need to sit downstairs at FIB only when I'm depositing or when I'm thinning down. But for pre-cuts, sometimes we can just do it more or less automatically.
Looks like Nesta has a question here. Do you have a sense of what the contamination you saw at 8 kV versus 30 kV was? Is it carbon?
No, that was platinum, because from the deposition.
But the contamination was the question.
Yeah, because this halo around, because maybe I can go there, because just a second. Here. So when I'm not sure. When we deposit, that's why when we do in situ, how I do it, after each time when I deposit platinum, I wait usually 20 minutes to 30 minutes before I start to image. Because platinum is a gas precursor, it's still in the chamber, and if you start to image too fast, it will fix on your lamella. It will fix on your chip. So this here is platinum contamination.
But I think Nesta followed up, it's not pure platinum, so.
Yeah, it can be. So in chamber, so platinum precursor is mixture of platinum and carbon. And then we have also gallium ions when we fix it. So it can be these three. But here, this is mainly, so it's platinum when we start to image too fast. And here I didn't. So here I was waiting the same as in this case. But at 8 kV, beam is not as focused as it is at 30 kV. That's why also here our contacts are not so sharp.
Interesting.
But I would really recommend, because that's why maybe also it takes a little bit longer for me. So each time, so for each step, when I start platinum, when platinum finishes, the deposition finishes, I will wait, even though the vacuum will, FIB will show that vacuum is already good. I don't trust it. I have serious trust issues with this. I still believe that there is some platinum in the chamber, and I will wait at least 20 minutes. If I have more time, for sure, I will wait half an hour before I will do the imaging. And I did notice change in the quality of samples.
Well, there's no question that sample preparation is still the most critical part and most overlooked part of TEM. It's and the one that we invest the least in. So it's
Yes.
just so critical. And the same goes for Richard. I mean, getting these tomograms. It's and also using the FIB method. As a specimen preparation is absolutely critical. I don't know if War has any comments or questions to ask. War?
Hi, Richard. Do you think in the area of the thickness that you can deal with, in each of the four methods you point out, they varies in the Z thickness, and in the case of the human platelet that you saw is very fascinating. So you could basically see all the organelle inside the platelet, right? Each of the platelet. And what are the variations from platelet to platelet? In terms of mitochondria or the other vesicles? Do they, in the same patient or in the same mouse, or the same physiological situations, do you see a broad variety of size distribution inside the platelets of those organelles?
Yeah, I think I think we've looked at quite a few cells, but sorry, I didn't quite understand that question. This was for the platelets, or which sample again? The blood platelet?
The platelet.
Platelets. Oh, okay, yeah. Yeah, we looked at lots and lots of cells. We see fairly consistent results, across different cells. One of the things is that these platelets, they can go into different sort of activation states, depending. Right. They're very sensitive to activation, so one has to be careful about that. And our collaborators use some special protocols to try to sort of prevent sort of activation if that's what they want to do. But it's interesting that in terms of the FIB SEM, what we found is that the gallium, has a very sort of serendipity type of role in how the FIB SEM works for biological specimens. If you wanted to invent a new technique for doing, high resolution, higher resolution imaging, you would sort of invent something that had something like a gallium metal, because the gallium needs to be an intermediate atomic number between the biological element, which is sort of doesn't give you any the carbon, nitrogen, oxygen, that doesn't give you any contrast in the FIB SEM. And it has to be intermediate between that and the real heavy atoms that you're using to stain it, which are the uranium, osmium, and lead. And so if you take the geometrical average of for example, of carbon and uranium, you sort of get something around the atomic number of gallium. So it in other words, it allows you to get contrast. You see the contrast, and at the same time between the gallium and the heavy elements, but you also prevent the prevent the electron beam from going too far into the sample. So this is an interesting sort of result that it just happens that the gallium is a good element for doing fibbing.
So you're saying that the gallium contamination, basically, the gallium.
Yeah, I mean, it wouldn't work without the gallium contamination. In fact.
Yeah, we hate it ourselves.
Yeah. But so if one, because was talking to some manufacturers, and they sort of are talking about doing fib-SEM with biological samples using a plasma source. But I would argue that, I mean, I don't know, I don't think it would work very well because it wouldn't give you any of the advantages of restricting the range of the electrons. So you wouldn't keep the higher depth resolution. But that's something that
I mean, that's very interesting. I think that's probably worth to look at systematically the thickness you fib it and also what you can see. I'm thinking particularly in the context of vitrified sample, not uranium stand samples. And some of the platelet actually can be pretty thin if you look at the edge of it, because we look at those, I don't know, maybe 15 years ago in the cryotomography, looking at the platelet, I realized fully the activated and unactivated difference. But it will be really fascinating to look at those in the fib right now and look at kind of low resolution distributions or the organelle inside the platelet in the active way, maybe inactive or different pathological state. So those are untapped territory as far as I know.
Yeah, and I imagine that in the preparation of lamella, using the fib to make lamella for cryo tomography of cells, that you're probably using a much lower, much lower amount of gallium, I assume.
Yes.
But I don't. It may be orders of magnitude lower, so therefore not producing this effect at all.
Yeah.
Does Vesna have any comment on that gallium for biology?
No. No, I'm also cutting biological samples, but yeah.
Are you cryoing or are you using cryo samples?
No, because I realize I'm mainly cutting teeth. I use much lower currents, and I realize that I don't have cryo possibility, but I see that if I use very low currents, I don't damage the sample, because if sample would be damaged, I would see in TEM yields would show immediately if there is damage. And I see that they are not damaged. But it takes always ages because I use really low voltages, low currents, and yeah. But I think one can do it without, I mean, at least teeth, because it's organic, inorganic.
Yeah.
This I can do without cryo. But of course, it would be nice to have cryo possibility, but I have to work with what I have.
Yeah, I think War's group has experience with cryofib.
Yeah, I think Nestor also has questions.
Hey, Richard, how you doing? Good talk.
Oh, yeah. Hi, Nestor.
A question for you on volume imaging specifically. Now, most of the techniques that people use for volume imaging are basically serial imaging techniques, okay? So what I was thinking about is, what if we still do your sectioning much like you did, but try to use a large parallel beam, not multiple electron beams like people have been talking about, but something like PEAMS, where we're flooding a zone and getting essentially a surface area image up to get a large volume. So now we can do more massive things. Think of the slicing technique, or the atom technique, but each slice, rather than going through an SEM, goes through a PEMS, where we're now getting an image of the entire slice or a very large area of it. Ever thought of that?
Yeah, I mean, I think I know that from what I heard that the Genelia Farm is actually working on something like that, where they have in order to do the very large scale, like mapping brain circuitry, what they do is they cut their blocks. I mean, I think these are still, you know, EPON embedded stained samples, but they cut them into thick slices, like maybe 1 to 2 microns or several microns in thickness. They put those slices onto a wafer, and then they sort of use a plasma, some type of plasma etch of all of those sections simultaneously. And so they gradually, you know, so they're able to get the 3D information out much faster that way, I think.
Okay,
so I haven't seen a follow-up. Yeah.
Okay. All right. Cheers. Good talk again for both you and Vesna. Thanks.
Thank you, Ness, for joining us, and thanks, Yi, for allowing that. So I have a class which I have to go to. So Kate, do you have anything to say to Vesna, and then Wah, please?
Yeah, I just wanted to say thank you so much to both our speakers. Vesna, I'm going to be putting some of those tricks into practice, and I'll follow up with you further. It was like so impressive how clean you got those. And Richard too, thank you so much. It was excellent talks on both sides. And Yi, thank you for all your service these past 3 years. We'll miss you for the rest of the EM-X, but thank you for everything you've done. You've been a great co-organizer. And then I'll pass it on to Wah.
Yeah, I agree, Yi. Thanks very much for helping us last few years and make everything very smoothly. And Richard, I really enjoy your lectures, and I will probably talk it remotely because I can imagine some of the stuff that we are doing in the cryo sample of the tissue and cells would can warrant some theoretical considerations what goes on through the depth of it. Again, Dr. Srot, thanks very much for making such a fantastically clean sample, and I think that I wish everybody have a great summer.
Right.
Well, thank you so much for inviting me to do this. I really enjoyed it, and so thanks a lot.